Translation is the process by which the messenger RNA codons are deciphered and a particular protein molecule is synthesized. And in order for translation to occur, we've got the messenger RNA, we've got transfer RNA, we've got amino acids, ribosomes, and also several enzymes. Now, the ribosome is a complex between the ribosomal RNA and protein. And it's got two subunits, a large one and a small one. And this is just kind of a matter of interest. Cells of higher organisms contain thousands to millions of ribosomes. That's inside the cells. So that's a lot of ribosomes. So you have these two ribosomal units, the small one and the large one, and together they form the ribosome. And I'm going to show you a video in a minute that kind of animates all of this. So here's an illustration of the transfer RNA. It forms this kind of a cloverleaf shape if you were to stretch it out and flatten it out. And so there's, I believe it's hydrogen bonds that are holding these guys together here. But it actually all kind of winds up, and it doesn't look like these little plus signs or something coming in. But up here is where it specifically interacts with an amino acid. So the amino acid interacts with an activator molecule and then reacts with the transfer RNA to form an activated transfer RNA molecule. So here is the activated transfer molecule. It's loaded, okay? It's got the goods here. So here we have the amino acid. This is the transfer RNA, and its function is to carry this amino acid to the ribosome as it's needed, to bring it. So there's a couple of steps in this protein synthesis. I should stand over here out of the, not blocking the screen. So the first thing that happens is the messenger RNA, and it's named that because it's the messenger from the DNA. It's bringing the message of this is what we need to put together, this is the order. So the messenger RNA attaches to the smaller ribosomal unit, and the first codon, which is always AUG, is going to occupy what's known as the P site. So here's the P site, and this is that initiating codon. Comes in here, and this is going from 5' to 3'. Then an activated tRNA with the complementary anticodon for that AUG codon attaches here. So that's what's showing here. That start codon codes for methionine. So the first amino acid is always methionine. That comes in here. And then the large subunit of the ribosomal unit comes in and attaches here. And now it's ready to go. It's brought all the pieces together and it's ready to go. The next step is called elongation. So next to that P site is the A site. And on the messenger RNA next to that start codon, let's go back here for a second. So here's the start codon. There's a codon next to that. This is what's going to be the A site. And yeah, I don't like that picture. So the A site is open, and the tRNA that matches up with that codon has an anticodon that matches the codon on that A site will come in and bind. An enzyme called peptidyl transferase is going to link the amino acid on the P site tRNA to the one on the A site tRNA to form a dipeptide. Then the transfer RNA from the P site leaves. It goes off to go get another amino acid. That was the initial one. It's going to go get another methionine. Like, okay, I came here, I docked, I dropped off my load, I'm going to go pick up another load. Kind of reminds me of like, a trucking industry. We're going to go pick up our load, we're going to deliver it here, they unload it, we go off and go get another one. So now the ribosome shifts along the messenger RNA. The one that was at the A site now moves over to the P site, and the A site is open for another tRNA to come in. And this just occurs over and over and over again. Translocation is when the ribosome moves down. So location, and trans meaning, like across, transcontinental. So it's moving. The ribosome is moving down the messenger RNA. It goes down 3 base positions at a time so that we can get a new codon on the A site. So here's the whole picture. So here we've got the ribosome, and we had this AUG start codon at the beginning. The tRNA comes in with a methionine and binds here. That's the initiation. So there's the P site, and next to it is the A site. So next, another tRNA that corresponds to the codon at the A site will come in and bind. Then we've got a peptide bond that forms. See how these tRNAs are holding these amino acids near each other? We're holding them together so that the enzyme can come in and hook them up. So they get hooked up, and the methionine comes off its transfer RNA. The transfer RNA leaves. This whole thing is going to shift down, and now here's the original starting codon, and that transfer RNA is leaving. Here we've got the one from the second, and it's got now a dipeptide on it. This site is, the A site is open, and so the next tRNA comes in, positioning these 2 amino acids next to each other, like this, and the enzyme forms a bond between them. And then this guy is freed from the dipeptide. He leaves, and the whole thing goes over and over and over again. And so it's this little factory, and it's like going down the line, bringing in the right amino acid and attaching it to the chain.
The chain gets longer and longer.
The chain gets longer and longer. This is why this section is called elongation. We initiate and then we elongate. And the protein that gets formed depends on what the code is that's on the mRNA. Some proteins are smaller, some are much larger. And then eventually it's going to come to a stop codon that will tell it to let go. The translocation occurs approximately 20 times per second. So you can imagine it, chunk, 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 20 times in 1 second. It's pretty fast. This transfer of this dipeptide to the next, from the P site to the A site, is called an acyl transfer reaction, because we're transferring this acyl group to this guy. And then the last step is termination. So the peptide grows through that elongation process. There are 3 different stop codons. And it'll encounter one of those on the mRNA, and that tells it to stop. Then the polypeptide is cleaved from the last tRNA that it was attached to through hydrolysis. Now, some stuff does happen after translation, some modifications of the proteins. All the proteins start out with a methionine residue at the beginning, because that's what the start codon, the initiation codon, codes for. And so that's usually removed. That's like you get little game pieces, and they've got these little tags on them that were just holding them together because they were molded plastic in a machine, and you take those off, right? They're not part of the actual thing that you need to use. So that's like a little bit of residue that's not needed. So it's usually taken off. There's a special enzyme that does that. Covalent modifications might occur. You may have disulfide bridges forming between the cysteine residues. So this is a protein, right? And this process of translation makes the primary protein structure, the sequence of the amino acids. After it's cut loose from the ribosome, Then we have the secondary and tertiary and perhaps quaternary structure that results, and that may involve formation of disulfide bridges, which are covalent bonds, and then there's going to be protein folding, depending on the alpha helix or beta pleated sheets, or however it is going to be structured. And if you've got a quaternary structure, so you have more than one component, then that's going to be assembled as well. So I think these summary slides are really, really helpful, because we can't really look at the chemical formulas of all this because it's far too complicated, and so we have to approximate them with drawings. So here's the whole business. Here's the transcription phase. So you have the DNA, and it partially unwinds, and you form the hnRNA, the heterogeneous nuclear RNA. And then that is edited. Remember, there's exons and introns. And the introns are interruptions, and so those need to get cut out. And there's more than one way that a given hnRNA can be edited. And so in that way, we can have one gene here that can result in more than one protein over there, depending on what's needed and how it's edited. So once it's edited, it becomes messenger RNA. That leaves the nucleus of the cell and goes out into the cytoplasm. Then the messenger RNA is involved in translation. So we've got activation of the transfer RNA, and those are going to come in to where the messenger RNA is attached to this ribosome, and it will do these steps, the initiation, the elongation, and this one goes over and over and over again, and then finally the termination and post-translation processing. Any questions? This is actually a very efficient process, because as this picture shows us, on one strand of messenger RNA, you can have multiple ribosomes on that same messenger RNA moving along. So it's a little bit like, like a water slide. So you don't have to go down one at a time and wait till the person gets all the way to the bottom. So here's a guy getting to the bottom. These guys are still coming down, and this guy is just getting on. There's a safe distance in between, but it can be generating more than one protein at a time. So many ribosomes can move simultaneously, and a polyribosome is a complex of messenger RNA with several ribosomes on it. So this is an illustration of a polyribosome.